there's been some debate on whether or not the uterus, the amniotic fluid, the placenta has microbes that are not pathogenic on a regular basis. And some papers were showing, yes, that's the case. Not huge populations, but they're there. And then another paper came out after that one, was saying, no, that was a mistake on your part. You must have contaminated the, when you took the samples, it must have gotten contaminated. And so there's still some debate about this, but I don't know. When I read the original paper, it seemed like it was well done. Of course, I wasn't standing over their shoulder watching them do it, but then another paper came along. This is a 2023 paper, and they're looking at maternal microbiota. This one, they're saying it communicates with the fetus through microbiota-derived extracellular vesicles. So we talked about these a while ago when we were looking at quorum sensing, nanotubes, extracellular vesicles. So you've already had this slide. I just grabbed it and put it in. So this is from that slide set where they know that pretty much any organism can do this. It's not just bacteria. Fungi can do this. Animals have extracellular vesicles, apparently even us. But they're finding that they can contain stuff, including DNA, even phages, other chemicals. And so they're finding presence of these in the amniotic fluid as well. So this is the abstract from that paper. And, they're saying it's limited information and it's controversial about the whole idea of this particular habitat. And this one, they said, extracellular vesicles secreted by the human gut microbiota have emerged as a novel mechanism for interaction. So they're tracking it in the gut, that there are extracellular vesicles that are getting released from one microbe and going out and perhaps interacting with another microbe. And so they were searching for the fetal case if that's happening there. And they said, yeah, in the amniotic fluid that they extracted from humans, so they had human donors, and they did have bacterial extracellular vesicles in humans. They also looked in mice where they had, it's much easier, of course, to get a larger sample size with mice. So they did pregnant mice and did the same thing, and they found the same idea there too. And so they're saying, yeah, that's, if you can find the extracellular vesicles, chances are the full bacteria are there as well. And so the debate continues, but this is another more recent piece of evidence that that particular habitat of the womb can be colonized. Let me see if I can stop share for a sec. I wanted to go back out. So then I ran across another paper. Let's see if I can pull it up. Let me do this. So this is looking at the use of extracellular vesicles as postbiotics. Have you heard that one? That term, postbiotic. So probiotic is living microbes, and you get that through fermentation products like yogurt, kombucha, things like that. Prebiotics are foods that you eat that the microbe can use. You don't really use it, but the microbes do. And so that's usually something like inulin. If you ever look on a package that says it has prebiotics, it's often inulin or chicory root. That's a common one. Or they'll say oligosaccharides. That's another common ingredient. And so those are prebiotics that feed the good microbes. And postbiotics is this, the way they're defining it here, they're saying it's a preparation of inanimate microbes and or their components that confers a health benefit. So now they're looking at the idea of using extracellular vesicles from microbes as a postbiotic health supplement idea. And so one thing about the vesicles is that they're really small. they think that they could be absorbed better by our cells, and the components inside could get easier to our cells and tissues. And so now they're looking at this idea of using extracellular vesicles as postbiotics. So I don't think anything's on the market currently. But they said this is an exciting area that we should be thinking about and could become useful for human health. So they say as the extracellular vesicles, they're inanimate. They can't replicate, so they're not dangerous. So if if for some reason, you wouldn't want necessarily a huge overpopulation of a particular microbe. And they're saying the vesicles wouldn't do that because they're inanimate. That's a hard word to say. Any problems with that? And so they're looking at this idea kind of early on in the stages, but look for that eventually down the line, maybe in a handful of years, they'll be selling things like this, or your doctor will prescribe things like this for you. Yeah.
So as far as like what would be contained in the vesicles, would that be like nutrients from the microbes or like plasmids with?
Well, they're actually saying, could we use this to like deliver drugs, like useful drugs to humans? I think that's what some scientists are really eager to think about. Like it could be a drug delivery mechanism that could be kind of easy to do. But I could, potentially there could be all kinds of applications that you could use that for because they're so tiny. They're made out of cell membrane material. So chances are they could fuse with our cell membranes. And so that drug delivery idea, it would kind of like allow for dumping in of drugs. And then, I'm guessing they could think about using it, they're studying it in the gut, like how are microbes doing in that in the gut, and could we use that to our advantage? I'll talk a little bit more eventually in the slide set about what do they call it? Precision. There's a phrase, it's a catchphrase. Precision therapy, something like that, where they're going at the idea of eventually we'll have enough knowledge where when you go to the doctor, they would assess your microbiome through a tool that's pretty easy, non-invasive, and then they could give you like a very specific recommendation based on your microbiome. And so, yeah, they're really excited about that idea rather than like we can go and buy probiotics off the shelf right now. And that's great. But they said we can even get better at this, where if a person is saying, I've been feeling this way, and they'll say, okay. And if they know the microbiome is influencing that, then they can tweak it. So that's the thought. And I think there are ways off from that, but maybe within your lifetime, as you get into, especially as you age, that would be nice to have that kind of precision health benefits. So let me see if I can extend. Okay. And then I think we went through this. So we talked last time about, let me reshare. Otherwise, it's not going to show up on the recording. We talked about this last time about the difference between C-section babies and vaginal birth, that they have definitely tracked differences early on, especially in the microbiome, doing fecal analysis. And then, of course, whatever that young baby's being exposed to right from the get-go, definitely that's going to influence their microbiome, their skin microbiome, as well as their internal microbiome. And they've done enough studies now where there's some general things they can say about once that child starts to develop and grow up, they start talking about this shift to the adult microbial profile. And basically, it kind of depends on what level of specificity. Like when they do these microbiome analyses, early on, they would do it, they'd assess what microbes are there at the phyla level. And if you think about classification schemes, phyla level is pretty broad. That encompasses a ton of different microbes up at the phylum level. And so if you want to get more details, you would go down to family level or class level or genus level. And so you have to take that into account. You have to look at the paper and say, how specific were they getting with how detailed in who's there. But basically, a lot of the early studies were looking at phyla, comparing phyla of bacteria. And they said that there's a certain profile that seems most humans kind of go into as they become an adult. And they say that these four phyla tend to dominate. And so I would like you to know these four phyla, please, because you see this over and over and over in the published literature. So the Bacteroidetes and the Proteobacteria are gram negative. I would like you to know which ones are gram negative and which ones are gram positive. And then the Firmicutes and the Actinobacteria are gram positive. So this is at the phylum level. This is very broad. Okay, so you have to appreciate there's a lot of species within each of these. Okay, so this is a pretty vague description. It's not super detailed. They're also saying that everyone is unique, certainly, especially when you get down to, let's see, like family level, genus level, species level, that fine of classification. When you get to that, they definitely can see we're all unique with our microbiome. We have our own little profile of microbes. Granted, we all seem to have these four phyla, but then when you get down to more detailed classification, there are definite differences. So we're unique, just like you've known you're unique in terms of the genetics that you were dealt when the sperm and egg united and made you, but now they're saying the microbiome is that way as well. So this is kind of a synopsis of this idea of going from birth to a few, maybe like 3 years old. And they're trying to show this idea that initially you don't have a lot of diversity. But then as the, baby starts to get more exposure to their environment, more interactions with other humans and animals, you start to see increases in diversity. And they start to name out some of the different phyla that are showing up over time. We'll talk about some of this in a little bit more detail. They talk about there's another area of study where they're looking at once the baby is shifting from just being fed milk to a solid food diet, so now they're saying diet influenced profile. And we'll talk more about this, but here's butyrate showing up again. We looked at that with the rumen and how microbes with, we were looking at the cattle or cow idea, their microbes are producing butyrate. So we'll come back to that idea. So dietary intake strongly influences. So we'll certainly be looking at that idea a little bit more. So they're saying in this one study, they're saying 60 to 70% were pretty stable as the, thinking about the process over the years. And then they were finding that certain phyla were a little bit more like buffered, like they weren't changing that much, whereas others were more sensitive to things that were happening with those adults that were influencing them a little bit more strongly. So the Firmicutes and the Proteobacteria seemed a little bit more sensitive to change in their environment. So we'll talk about that too. We'll look at how microbes that especially in the gastrointestinal tract, how they can be influenced by your activities pretty profoundly, actually. This is kind of a nice visual. So these pie charts essentially are with the different colors are showing, this is at the phylum level now, those four phyla. And then others is the gold. So changing over time in response to varying environmental conditions, life stages. So in this paper, they were not indicating in the fetus that there's anything going on with microbes. So again, it's controversial, but at least the papers I've read, I'm fairly convinced that there are microbes that are normal microbes hanging out in that area of the womb. But anyways, so you can see if you just kind of scan slowly from pie chart to pie chart from left to right, you can see the color differences, microbial differences. And they're looking at both, healthy conditions as well as obese or malnutrition as you get older. So what happens elderly, looking at those pie charts compared to the ones further to the left, is there one thing that you could say about those pie charts? Yep, exactly. Less diversity. And so this is something that they're tracking, with elderly health. This is kind of a hot topic right now, looking at, and we've talked about this through the semester. The idea of high diversity seems to be a good indicator of a healthy ecosystem, whether you're looking at a forest or the microbiome of a human. And so again, this idea of less diversity and what can we do about that to kind of tip it back to a younger profile. They've even I think most of the studies are in mice, but they've even done fecal transplants where they can show mice that get a young mouse's fecal transplant end up with higher diversity. And so that was a very interesting study where they were showing that perhaps that could be a strategy somehow in the future. But probably most of you know about fecal transplants, I would guess. That's actually not a new thing. They actually would kind of do enemas of fecal transplants. They would do it through the enema route, and that was actually practiced quite heavily in veterinary medicine. Back in the day, I talked to a veterinarian who did large animal practice, a couple decades ago, very active, and she said, "Oh yeah, that." And when I was really happy to chat with her because she said, "Well, yeah, because you're essentially putting in a bunch of microbes to outcompete the pathogen. And so you're like flooding the system with good microbes from healthy." I think she was talking about cows. She was working with cows at that time and horses. And she said that that was a common strategy. If they didn't know what else to do, they would do a fecal transplant and often the animal would recover. And so it's not that new, but doing it regularly for human health is fairly new. But there's been case studies where fecal transplants have allowed, or this one case study of a woman who got very, very sick with a pathogen and none of the antibiotics worked. So then her husband donated fecal material and they did the enema transplant and she bounced back within 48 hours. She was back to normal when everything else was failing to help her. So these types of ideas, of course, that's not a very attractive health procedure, but if it's going to save your life or make you that feel back to normal, it's worth it. But companies are popping up now where they're doing fecal transplant ideas with swallowing a pill rather than an actual fecal enema. So they're coming out with other strategies besides that method of application. Skin microbiome as well as internal is something to think about. So of course, your skin is your most direct interaction with your external environment. So as you move through your day, you of course are interacting with microbes over and over and over and over, including microbes that are in the air, floating around that you come into contact with. So it's not just solid surfaces that you're coming into contact with where there's microbes. So there's a New York Times article with the title, "Trillions of viruses are raining down on us every day from the sky." And that's accurate. Most of those viruses have nothing to do with you, though. They're just environmental viruses. So you have skin microbes, you have internal microbes, of course. So your whole body is well colonized. It's not, a lot of the studies are focusing in on the gastrointestinal tract, but don't forget that we have all these other areas. And again, nice pie charts showing the differences here. You can clearly see that depending on the body part, you see lots of differences and pretty decent diversity. This is not, of course, an exhaustive list. So they're even showing in the stomach. So that's another controversial area that has been debated quite heavily. But yes, there are microbes that hang out there, surprisingly. So let's get into that a little bit in terms of some of the terminology that you see when they're describing these different microbes. So most of the microbiome research, what we're thinking about is the resident microbiome. So those are the ones that are hanging out with us long term. And then you have transients that you come into contact with, but that doesn't necessarily mean they're going to persist with you. So the word transient means that it's, yes, you might pick them up somehow, whether it's on your skin or you eat some of them and they get into your body. And they might persist for a while, but then they tend to fade away. So transient for that idea. And then the opportunists are typically the pathogens that somehow gain a foothold and can take advantage of that. And so this is usually when you're thinking about if you're sick, sometimes you can get a secondary infection. So let's say you're sick with a virus. Antibiotics for viruses aren't going to do you any good, by the way. But you're feeling bad, your immune system's compromised, so then it's easier for another pathogen to come in and take advantage of that and set up camp on or in you and make you even more sick. So that kind of idea. Also, for example, if you go in for surgery, there have been too many case studies showing that microbes can get introduced during surgery from one part of the body deeper. It's often deeper into your body as they're cutting into you or manipulating your body during the surgery. They also note that with extreme trauma, like if you get into a car accident, you can have introduction of microbes into novel places of your body as well. And then if you do something that disturbs the normal microflora, so one huge disturbance would be, we've talked about this, antibiotics, right? So if you take antibiotics, sometimes you have to. I'm not saying you shouldn't take antibiotics. I'm just saying if you do that, and sometimes it is necessary, you have to appreciate that it is going to disturb most likely the normal microflora because most doctors give you broad spectrum antibiotics, which is different than narrow spectrum. And so if you go to the doctor and he or she is saying, yep, you're going to need to take some antibiotics, maybe you should ask, hey, is it broad spectrum or narrow spectrum? And get into a conversation about that. Because if you can, you want to request narrow spectrum. The problem with that is they have to know what you're sick with in order to do that. So they have to be very confident that, yep, you got strep throat. Then you could say, okay, give me a narrow spectrum for that. I don't want a broad spectrum. But if they're like, yeah, we think you have a bacterial infection, but we're not sure, then they're going to give you a broad spectrum. And so that's often they kind of hedge their bets and go with the broad spectrum. More doctors are becoming aware that that might not be the best strategy for your overall health, though. And so I think if you bring up that conversation with them, some doctors will be aware of that and will be able to converse with you and help you. Maybe they can come up with a narrow spectrum. Have you heard that narrow broad before? That's something that I hope sticks into your head, because I really do think I had a previous student who took this class and he took general microbiology. He didn't do it for himself. He did it for his beloved dog, was very, very sick, and he took it to the vet and he had that conversation with the vet. And she's like, "Are you a pre-med student?" And he's like, "No, I took microbiology." So, but she said, absolutely, you're on the right track. So she was a very well-educated vet. And so she did prescribe a narrow spectrum. The dog recovered and was able to get back to health. But this idea of trying to keep your normal microflora untouched and just target the pathogen, that's the best situation, but sometimes that's not possible if they don't know what you're sick with. So when I took microbiology a while ago, there were many sites that they said, "Oh, that's sterile, like the womb, the uterus." But now more and more they're finding microbes everywhere, including the brain. There's indication that there's microbes hanging out in our brain tissue as well, and extracellular vesicles. They're finding them there as well. So lots of interesting research going on there. That's your tongue, or a tongue. That's a human tongue. So all the different dyes are different groups of bacteria primarily. So this is a study that they actually did where they were like mapping out the ecology of the tongue, like who hangs out where, are they going to certain areas, setting up camp preferentially in certain areas. So this is like the middle of the tongue. You can see that there's less microbes right down the middle line. And they were even looking at colonization strategies, like where do they colonize first? And so this paper, they were really getting into it. So the dentistry world is getting pretty excited about this information because it's really showing how oral health, obviously it's very important for your mouth, but they're also showing that some of these microbes can migrate to other parts of your body, especially if you have oral surgery. And that has actually caused some problems. So there's a lot of interesting research going on with that. So think about that next time you kiss someone. Especially if it's a nice deep kiss. Yeah, you got a lot of swapping going on there. So that brings up this idea. So this is an idea that's been floating around for quite some time. I've gone to conferences over a decade ago, and this was something that was well known back then. So it used to be called hygiene hypothesis. Now they're calling it old friends hypothesis. And I'll show you a little video in a second here. And so this is the idea that the normal microbes that hang out with us are largely beneficial, or they're not really doing any harm. One or the other is what is generally seems to be the consensus. And they say that if you live in an environment that's super clean, well, then you're not picking up the normal microbes that coexist with us. And that might actually be a negative thing for your overall health. And so when my kids were babies and toddlers, that was the era of antibacterial soap, antibacterial toys. They would impregnate the plastic toys with antibacterial compounds. And it was just, everyone was on this kick of everything's got to be sterile. That baby has to be in a pristine environment. I did not adhere to that because I had microbiology training. So yeah, we didn't have a lot of play dates, I guess. But yeah, my sons are healthy, so I don't know. But this idea that, microbes are evil and they're bad for us, that was a real thing for a long time. And unfortunately, it's still a thing among people who don't have any knowledge of microbiology. It's getting a little bit better, I've noticed, compared to a decade or so ago, but they're starting to link this, or they have linked this idea of ultra clean environments and this correlation between that and increasing rates of allergies and asthma. And they started to ask the question, is it because these people, these kids, aren't getting proper exposure to all these different microbes? And they also show that it definite differences in the gut microbiome of these people that were exposed to really clean environments, and not having that diversity of microbes around them. They seem to have issues with their gut microbiome and also things like allergies and asthma. As the hygiene hypothesis, have you heard about that anywhere? Yeah.
I actually got really, really sick from American food when I came back from India. Ended up getting hospitalized and stuff, and they said, yeah, our food is too sterile. And when you were over there for so long, you changed your gut microbiome,
Mm-hmm.
and it ended up causing a bunch of stomach problems that took months to actually recover.
Right, yeah. Yeah, that's a huge shift coming from eating, that type of food. Oh, yeah, I could see where that would be challenging. So, yeah, it's amazing. They're also showing that too. I don't think I stuck it in this slide set, but I'll put it in there. There's a nice study showing several different geographical locations, and they're showing clear differences, in what's there and the ratio of what's there. So, yeah. So this idea of, being too clean, it makes me think of Pigpen, right? From Charlie Brown. So this idea of being too clean, let's see if I can pull up that video clip. Let me stop share. So let's see. Do this. So this is that documentary with Liam Neeson narrating it.
How do you think more work to clean? Consider the research of pediatrician Erika von Mutius. She treats allergies and asthma, conditions in which the immune system overreacts to harmless substances. Rates of both disorders are on the rise in affluent, industrialized regions. Perhaps children are growing up in surroundings too germ-free for their own good.
Microbes do a lot of harmful things to us, but they may also be important for our immune system to learn how to deal with the environment and how to tolerate and fight viruses, bacteria, and infections.
To understand the causes of allergies and asthma, von Mutius is conducting research in a place where these conditions are rare: the Bavarian countryside. She wants to sort out exactly which environmental factors may be protecting children who grow up here.
The study we're doing is a comparison within little villages. So we compare children who live on a farm to children in the same village who do not live on the farm.
She has enlisted over 800 families with children between the ages of 6 and 12 to participate in a detailed survey of health and lifestyle.
Dann wie viel Zeit ungefähr pro Tag? In this questionnaire, we asked for allergic conditions, and then most importantly, we asked for the contact to farm animals and farming activities. Sie haben Pferd, Schaf, Ziege.
Her goal is to create a profile of environmental exposures for each child. The team analyzes dust samples from carpets and bedding throughout the house for the presence of animal hair, dust mites, and microorganisms. If the family keeps livestock, samples from the stables are screened for microbes released in the shedding and droppings of animals. The study is in progress, but preliminary results suggest one very strong correlation.
One of the factors that seems to be important is the contact to the livestock. That these children, the more they are in the stables, and the earlier they are in the stables, that this gives a protection against the development of allergies.
High levels of microorganisms in the stables may help prime a child's immune system for life.
Microbes have been around us always, and probably we need to find the balance between eradicating the harmful effect of bacteria and maybe also taking the beneficial components of this. But this is really into the future.
Our species evolved in a world awash with microbes, crowded with other creatures. We've only begun to understand the value of this heritage.
Scientists and medical researchers who focus on the subject are more and more in agreement that it's a big mistake for humanity to separate itself from the rest of the living world too much. The vast majority of species out there are friends. They're not our enemies. And we not only benefit from them, but as a whole, they are essential to our existence. We're the fortunate heirs of more than 3 billion years of evolution that created this stupendous diversity. We need to learn a lot more about the living world and the way that humanity itself is affecting evolution.
Like all living things, humans are a product of evolution, but we are the only species that knows it. We alone can see into the distant past and marvel at the history of life. We alone are beginning to understand that we can use evolution to shape the future for all of life. More than anything else, this unique vision may be what makes us human.
Okay. So this, keep in mind, this was made quite a while ago. So it looks like 16 years ago this was posted in here. So Erica von Moodius has published many studies since then. I've been off and on keeping track, and she's still doing this kind of research, and other scientists too have come in and added to this body of knowledge. And they're definitely supporting that hypothesis that the ultra clean environment is not a good idea when it comes to basically our immune system needs to get a workout. It needs to get that challenge of all these different microbes so that it can, get practice, so to speak, at figuring out friend versus foe. And they think that the ultra clean environment is not allowing for that process, and that can lead to some dysfunctional immune system issues. Has anyone taken immunology? Does Dr. Sharp talk about this idea? Do you remember? With does he call it hygiene hypothesis or old friends theory? Okay. Yeah, there's another video I posted let me go back. Well, let me just show that. It's just 3 minutes. Let me just do that real quick. So now they're starting to call it the old friends hypothesis.
Can you tell us a little bit about the hygiene hypothesis?
Sure. This was a hypothesis that was put forward by somebody called David Strachan in 1989. And basically, he came up with this idea to try and explain why we saw so many more people getting allergies in the 20th century.
What do you think is the role of the world around us in changing how our immune system works?
So what Strachan noticed was that in the country, in particular farms, people tended to have less allergies than people who lived in cities or towns. He also noticed that people who were in very large family groups, so a lot of brothers and sisters, were also less likely to have allergies. So he wondered if it was early exposure to germs that was really protecting people from allergy, and you were more likely to get that exposure if you had a big family or you were living in the country.
So what are some possible explanations for this?
Well, the idea would really be that the early exposure to germs is educating your immune system and giving you a fit immune system so that it doesn't start responding incorrectly to things that should be harmless. Because basically what an allergy is you reacting to something that's completely harmless that you should ignore.
So is it all germs that we should be thinking about here, or are there some specific ones involved?
So that's a very good question. There's been a lot of research on this, looking at all sorts of different bacterial infections and also viral infections. And the research has basically produced very conflicting results. They're just not clear. However, the hypothesis has moved on to focus on the old friends hypothesis. That introduces the idea of parasitic worm infections and their role in perhaps protecting us against allergy.
So your own work is looking at parasitic worm infections. Can you tell us a little bit about that?
Yeah, we are very interested to understand how your immune system deals with parasite worm infections. And in particular, we're trying to understand the products that parasites make and how they manipulate the immune response to promote their own survival. And lots of other groups are doing that as well all over the world. There's even clinical trials using parasites to try and prevent disease. The other thing that we're trying to do is we're trying to understand why some people have a very bad immune response to worms, because obviously you don't want to give somebody like that a worm infection. So we're looking at biomarkers that will help predict the people who are less likely to do well with worms.
So yeah, this is, I read an interesting book on
As
this
I'm in the midst of.
concept of deliberately ingesting parasitic worms and letting them set up camp in your body with the goal of this person who wrote this book. He had a severe allergy, like it was definitely impacting his quality of life, where he was miserable. And so he decided to give this a shot. And so he wrote a book about, you know, trying to find people who would allow him to do this, do it safely. This was published a while ago, so it wasn't a very well-known idea. It was kind of a fringe science at that time, but it's starting to get looked at more carefully and thoughtfully. So that could be a thing too for future health issues if you have allergies, especially if they're pretty severe allergies. So stay tuned on that one. We'll see. Okay, I guess we got to go. So I'll continue on with this on Monday. So those videos are posted in EDUCAT in the readings and videos section.